Okay, for people who are joining this live stream, we are talking about biological sequence processing in Wolfram Language, and we have both a bunch of internal folk and some outside experts who I hope will help us and me understand what is going on here. So, what is the basic idea? So, the basic idea, oh boy, that's a lot of functions to do with biosequences. Really?
You had said the last time the human genome will never change substantially. You might well have 20 functions to deal with it.
Oh God, you took me literally.
Yes.
I mean, usually in a lot of these things, we're trying to have sort of named, string named properties and things like that, rather than so many explicit functions. All right, but okay, so let's just get some sense of what this is. So biosequence representing, so essentially these are long stringy molecules of the various kinds that biology chooses to use. Is that?
That's right.
Okay. And so what is a sequence molecule different from a biosequence? That's something which is the molecule representation of a biosequence?
That's right. That's the function that takes a biosequence and will return a molecule with the appropriate end caps and so forth.
So there is in a sense, you should also be able to take a molecule, and it's like recognizing, a particular pattern of molecule should be recognizable as a biosequence of a particular kind.
Absolutely.
And
yes,
that function. Yeah,
that's a harder job than going the other way.
Obviously, right.
So we haven't got that set up yet.
Right, but okay, that's like digesting, here's all the atoms in a lump of RNA, tell us what the RNA sequence specification for that should be. Okay, this seems like an excessive number of functions, but okay, let me just understand what, okay. So I see, so things like this and blast-like things, is it the case that there's a version of similarity that is different depending on what biomolecule you're looking at?
That's right. Depending on what kind of biomolecule there is, they can have different degenerate terms and therefore different similarity calculations.
Okay, but so just so I understand, and this is a question, maybe Trevor or somebody knows this. Blast and things like that are a reflection of the physical propensity of different bases to change, or are they something that reflects the historical likelihood of bases changing in the way that DNA is actually used by biology?
This is Trevor. It's a bit of both. So that kind of encoded, if you look at, say, a PAM similarity matrix or a BLOSSOM matrix, which is what often will be fed into these things for amino acid similarities, that will rely on like looking at, you have evolution that's happened and things have diverged from one another, but it used to be the same thing. And so kind of encoded in how much similarity there is both some process of how transition kind of bias. So some mutations will happen more often than others, but also kind of amino acid similarities, so that some amino acids will substitute more easily for other amino acids and won't be rejected by natural selection.
But so when we have a biosequence and it's a piece of RNA, for example, do we have to know something about this RNA was in, I don't even know if they have it, an archaea versus this RNA was in something else to know what the correct similarity?
That's a good question. So if you wanted kind of an optimal alignment, you would probably want to know about that the most important parameter for RNA would be this transition-transversion ratio, AG versus CT. And that would, if you had like a long enough string, you could learn that yourself, or you could kind of have some simple kind of default that would more or less work for that. It's not going to be hugely mattering, but it will matter if you have you can imagine a little segment that is like A's and G's versus C's and T's, that you might align it slightly differently if you think that A's and G's mutate together more often, mutate to one another more often.
Okay, so the bottom line there is there probably needs to be some provenance annotation for biosequence to help, or some at least metadata for biosequence to help with things like this. Is that
yes, ideally, though it seems really hard. That
okay,
you're gonna probably want to learn it from the data, that rather than like coming in and having a lookup table, whether you're archaea or bacteria or mammal.
Fair enough.
Yeah.
Okay. Okay, so this is going to be a can of worms, this whole business about whether we now, I mean, there must be some very naive things here, like the translation from, I mean, and this is stretching my pathetic biology knowledge, but I mean, the very, like the various translations from RNA to proteins, and the U case for and again, I'm going to embarrass myself with my biology knowledge, but when there's a U instead of a T and things like this. So presumably I mean, do we have some functions here? Oh, I see, we do. Say
there will be examples of reasonable depth in each of the sections. That was just for a very quick overview.
Okay, fine. All right. How much of this actually exists, by the way, John and others?
Everything that's in black exists in the prototype build. So everything except for the finding, the gene positions and genes and SNPs exists in the build. The finding does exist, but it's waiting on integration into our infrastructure for the databases and that kind of thing.
Fine. Okay. So then, okay, and this is using the standard conventions for what W's and things like that mean in terms of unknown, undetermined between different bases type thing. Okay, cool. Okay, so if I type this in, I mean, so if I just say random choice, let's be I don't know, I think these are crabs do this, don't they? Where they have a lot of if I just say this and then I say string join, does it work for the list of symbols as well? Probably should. The biosequence can take a list as well as taking the string.
I think it does just take the string right now, but I'll make it take the list for sure.
Okay, why is it? Oh, I see. It doesn't like that because I didn't tell it what it is.
You need to type.
Okay, that should be better error messages rather than just sitting there for a long time and being confused. Okay. All right, fine. Okay, what can I, well, I'm probably going to ask in a minute what I can do with this. Okay, an RNA, amino acid. I assume that these are all totally standardized at this point.
That's right. Yes. I mean, we support the standard ones, and the discussion of whether we want to override types for non-standard amino acids, that's a potential topic for discussion.
But I mean, like somebody who's building their own synthetic biology that has some funky, I don't know what, germanium-based life form or something. No, those don't exist. But a funky amino acid. Is there some standard way of representing that in one of these strings?
No, generally not. Generally, it's limited to the typical.
Okay, but I mean, for the biology folk on this call, I mean, like for example, for us, could we do it by like having a dash there and then having some key or something? I don't know how it would work. I mean, or having some Unicode character there that represents something different, or insisting that if it's non-standard amino acids, that this thing be a list rather than a string, and then being able to put some symbolic object in there, like a whole molecule, for example. I mean, I suppose that would be the most obvious way to do this, right? If it is to insist that this be a list, to explode this into a list, and then to have just a single character, G or something there, to just be a happy, ordinary amino acid. But then in general, in worst case, to have an actual symbolic molecule object there to represent kind of, an arbitrary amino acid. Is that crazy?
Stephen, we have something similar where we have to deal with conjugates between biosequences of, like it's an amino acid connected to a piece of DNA. And sometimes there's something synthetic in the middle, like a weird like remnant from a click conjugation or something like that. So we actually ended up creating like a slop thing that was biosequence. Ours would be sequence, but it was like what do we call it, modification or something?
Yeah, modifications. You can just kind of put whatever you want that you define to be custom in there. Like if you have a fluorophore at the end of a piece of like DNA, you just
it's a little bit like the user-defined unit thing, something like that.
Yeah, like an independent unit kind of thing.
Yeah,
I see. So okay, but so that then in this biosequence, what you're saying is it is convenient to I mean, if we made this into a list of just string characters followed by some symbolic thing that represents the weird thingy that's hanging off the end, that would be a reasonable way to do it. Is that what you're saying? And that would be somewhat consistent with what you have built. Is that a true statement, or is that far away?
That or some larger wrapper around a list of biosequences that say these guys are all in a line. That's how we solved it. We have like a conjugate, we call it like a strand.
Yeah.
And then there's a strand is a series of sequences which can include these weird chemical modifications that are unnatural.
So then you might have biosequence. Okay, so let's say that biosequence was a thing that could be nested. So it's biosequence of biosequences. I mean, that's a little
and
odd.
we do allow this.
Okay, but okay. So then what it might be is an amino acid followed by some weird piece of glue followed by another amino acid. Is that the idea?
Or a piece of DNA. Yeah, exactly.
Yeah. For me, my level of this stuff, it's okay. All right. So find. Okay. So find a SNP. That's a very weird return. That's very weird.
I guess what it's saying is at the 569th position, you can find that.
I'm well aware. Yeah, right. Yep. I'm well aware. But none of our other functions that find positions of things work that way. I mean, sequence position, for example, would find. I mean, what sequence position would do is to return the starting and ending coordinates. Let me understand something. In the world of snippery, I mean, whenever I play around with my own genome and I want to find some particular SNP in my genome, I do the incredibly Neanderthal thing of I find flanking sequences and I just go search for those literal flanking sequences in my genome, because that way I don't think I'm going to get it wrong where the SNP position is. What is the official definition of one of these SNP positions? Does that make sense as a question? I mean,
so
maybe somebody does maybe Trevor or the Emerald folk have a comment on this? Did my question make sense?
Yeah, it did. I don't know it for these RS numbering because I don't work in human genetics enough. If I was doing this with anything bacterial or viral, I would need basically, it always relies on a reference genome, and then it's position 500 in this reference genome, and you have to have supply a reference.
I see. And so then once you've got that, so then the analog of a SNP position for you is with respect to this reference sequence, it's position 20,000 or something in this reference sequence.
Exactly.
And then when you go line it up, then you know that there'll be many changes from the reference sequence, and then you actually have to have code that goes in and finds flanking sequences and things, or however that works, to try and line up position 20,000.
Yeah, that's relied on an alignment where you align it to the reference sequence, or do align multiple things to the reference, but the reference gives you that fixed frame that you're going to.
And what happens if it isn't? What happens if it's undetermined? I mean, so in other words, in practical terms, if there are multiple places, if the thing is a mess and it has a bunch of repeats in it, what typically is done?
People throw up their hands.
Okay, fair enough.
Case, yeah, and that it's low complexity. We don't have a definitive alignment.
Yep. Okay. The way that this one works is it finds the best available position against the reference, and then against a fairly broad nearby region takes the alignment between the given sequence and the reference sequence, and then will search for the SNP difference, kind of where it's supposed to be given the corrections from that alignment.
Wait a second. So, okay, so hold on. So you have an algorithm that basically says, given, I mean, this SNP you could go look up in the reference human genome, and you could know all its flanking stuff and so on. I have no idea what happens as you go between two different reference sequences and things like that.
It's hard, but the way I handled that case was I would look for nearby genes between different genomic builds and use that to kind of recalibrate where the sequence.
Okay, but I mean, this can really matter. Like, I mean, you know, you absolutely cannot get this wrong, right?
Right.
I mean, so, I mean, this is going to be, you know, big deal for. Okay, so actually, Emerald folk, have you had this issue as well or not?
So we don't do a lot of bioinformatics right now. We're mostly useful because we do bioorganic chemistry. So previous section we do a ton of, not a lot of this yet. So sorry.
Okay, fair enough. So you don't care yet. Okay, all right. Okay. So I understand. I'm a little scared of what you're saying, John, and I think we need to look at that very carefully because we really don't want to make a mistake. I mean, much better to say we can't find this SNP than to say, no, it's here. You know, you've got a T for that value, and then it turns out that the person's genome is actually a mess and they don't really have a T there.
I agree. Yeah, that makes sense. We should have some really great testing for this and review for sure.
Okay, I'm also not thrilled about the return value here, and I don't really like this, but let's see some more before we come back to this. I mean, I understand, I think this is the wrong way round. I think it's, I mean, this reminds me of text cases, for example. Have you looked at the design of text cases? I mean, in other words, in text position, for example, let me start up let me not tempt fate. Let me use a 12.0 here. So in text position, we've got a similar kind of thing. Find, okay, so in text position, what we might do, yeah, there we go. So this is, we've got various annotations for how to present what you find, right? The start and end position, the string identified, etc. So I could imagine the string identified is what you want here. You get what I'm saying? So in other words,
yes.
You say, this SNP, what is it in this case, right? So I think it's better to have a design more like, you know, a biosequence position or something like that, than, right? But let's do a light look through all of this stuff, and then we'll come back and get into more details probably at a different time here. Okay, so because yeah, I would like to know what is at that position, you know, and this SNP, I mean, are all the RS SNPs single positions? Are they all single? You know what I'm asking?
No, not all of them are. No.
What a mess. What else are they?
I mean, they can be longer, and there can also be kind of multiple variants identified per. So it's kind of one standard of arbitrary length, and then the multiple variants that are associated with it.
Okay, but if we look at the properties of that SNP, for example, we will be able to find out, in general, you know, what you know, what the different, I don't know what is the property called? That you know what I'm asking?
I do know what you're asking. I don't remember offhand.
Okay.
The alleles that are involved, maybe. So this is, I just looked this up. This is AC to AG, or as the SNP.
Right. So what this is saying, weighted allele frequencies here. This is not the world's most lovely representation of that, I have to say. Okay, so that's just showing, because that's what we're typically going to want, right? I mean, given what it's going to want to say is, okay, in this particular biosequence, the result was an A there.
Yes, there's also later an alignment view that shows this detail.
Okay, fine. All right, so let's take a look here. All right, find the locations in the corresponding to genes. Wow. Now, isn't that kind of complicated in general? or is that now considered a solved problem? I mean, if I throw you an arbitrary random, RNA of some random virus, is it even, again, biology question, I don't know the answer. Is it even well defined where the genes are?
Only if you have, I'm assuming here, you have a lookup table of like, you have all of these hemoglobin sequences and you're using BLAT or something like it to do a very fast alignment, or not even real alignment, a very fast kind of seeding sort of alignment to be able to say, yes, these are identical. I don't know what happens if you have some minor variation and then things start to stretch a bit, where if you have a bit of diversity, maybe it's hard to kind of make that call.
Okay, but I mean, naively, a gene is sort of a thing, a piece of the sequence that gets transcribed and turns into something. Mm-hmm. But if I understand, I mean, it used to be the case, I don't know whether this is a solved problem, like 15, 20 years ago, it used to be like, where do the genes start and stop on a genome?
Yes. So this will, behind that gene, behind hemoglobin subunit alpha 1, you'll have all of these different forms, these like different splicing forms and so forth. So like one gene might have a locus in the genome, but they'll have a whole bunch of different RNAs that are associated, that are kind of created by that gene, which creates a bunch of different proteins that are different kind of splicing variants and so forth. So there's definitely complexity there.
Okay, but there's a different thing that we could ask for here from this, biosequence features, which is use some heuristic to decide where, I don't know, coding sequences or something start and stop on this particular, like for example, can I ask, where are the coding sequences on this biosequence?
It's a reasonable question. It's not in this implementation, but that's a reasonable thing to ask.
But isn't it, I mean, that's a sort of machine learning type question, isn't it? I mean, there's not a precise answer to that. Is that a true statement?
Yeah. Go ahead.
Yeah, there's the distinction between the ones that are known and the ones that we could potentially determine through something like ML.
Yeah, right. Yeah. That's right.
Sorry, go ahead.
No, I mean, it is my vague impression that the state of the art is quite decent on determining where those things are. Is that a true statement or not?
I believe so. So all of the ENCODE data has done a bunch here. You do have alternative start sites that crop up often. First, that's like, there is the rule, and then there's the exception to the rule, but you can actually figure out what letters that were upstream of the promoter caused this exception to occur. And so, so you might need more context than that would be provided by just the biosequence. Like if you were missing kind of the stuff that's a bit upstream, you might not be able to make the easy call, for example.
Fair enough. Okay, so what we're seeing here, what on earth is this in this entity instance, this reference sequence-based pairs? So that's saying these positions in this particular biosequence are this subpart of the hemoglobin subunit alpha 1.
That's right.
Is that right?
Yes.
Yowie. Okay. I guess biology is kind of complicated. It's what happens after a couple of billion years. Okay. And what are we seeing? Okay, well, I think we should keep going and just do a quick scan through everything here. Okay. What on earth is this? So this is showing SNPs and alignment.
Right, so this shows where in the alignment the SNPs are found.
But this is an alignment. How did it end up with two sequences here? In other words, this thing is saying that there were two sequences and it's got a comparison. There was a T in one and a C in the other. But
right, it's the one provided is the first and the second is the reference sequence.
How does it know what it's a reference? I mean, how does it know if this is a horse or an elephant? I mean,
human is assumed throughout. So this has all been human.
But why? Okay, but I mean, that doesn't seem self-evident that that's what we should do. In other words, wait a second. So the SNPs obviously are human SNPs, right? So right. I mean, we can imagine some kind of dollar default organism or something,
right?
When the apes take over, they can change the default organism. I'm sorry. But nevertheless, in a case like this, I don't understand why you're. Oh, I see. Alignment is another thing. Is that a third argument, or is that a?
That's a third argument.
That says what to display in the output.
That's right.
Find the SNPs and display it as an alignment.
Yeah. So
okay.
The third argument is the result format.
So that's similar to the thing that I was complaining about with text position, although the way it works is to use a rule to specify, it would say SNPs arrow alignment in the way that text position works, for what it's worth. Okay, but
that's a good convention. Yeah.
Well, we should come back to this. I'm not sure it's the right thing for this. Okay, cool. Okay, classification properties.
Can we ask a quick question about at the bottom level, is there a way to handle degeneracy in the sequences, in the biosequences themselves? To say that this position is like a Y or something?
I think that's what we saw earlier, yeah.
Okay. Yeah, absolutely. All the degenerate bases are built in, and there's a fair number of functions for processing degenerate bases. It's also one reason why we have a different sequence alignment, is to support degenerate base matching.
Got it. Okay, thanks.
Okay. Oh, I see. So wait a minute. So sequence alignment has been generalized to deal with degenerate bases. Is that correct?
That's right. Or if you in the similarity rules option, there's now a use degenerate bases value that can that allows it to identify them.
Okay. All right. What's all this stuff here? What on earth is this?
So this is similar to classify, and the problem is that Trevor was telling me that frequently you don't only want to know what the potentially the best match for position is, you want to see all of the likely matches.
Mm-hmm.
By for a position, you mean, I don't understand what that means.
So
go ahead.
So given the mutations that might occur, it could be that the sequence you've provided could be mapped to multiple standard positions on the genome, and so in order to not just see the best one, but to see all potential results, we've exposed those underlying probabilities in the same way that classify does.
Okay, that makes sense to me. I mean, this looks like kind of a mess. I mean, the way that the way we're specifying this position on the genome, I think we're going to need a wrapper for that. I think this is that seems too. I mean, really, what that is essentially a what is it? It's like a biosequence location or something. I mean, it's analogous to, it's the analog of geoposition, I suppose.
Ooh, that's good.
Not really, because here we have an entity and then we take a subpart of it. With geoposition, we don't have other entity for Earth. We don't make a geoposition relative to a continent.
Okay, let me try and counter that. I mean, so geoposition with a geomodel our moon is certainly referencing, its lat long is very different from the lat long on the Earth. I don't quite get this. I mean, you're saying, my point is that geoposition, admittedly, it's real numbers instead of integer positions, is something where it is relative to an existing object. We're saying, what position are you at?
Right.
And this, I mean, okay, I understand this strand specifier. I mean, this is saying on the Earth, that's chromosome 5, or on I don't know what, random asteroids, Ceres. This is the location that we are talking about on the asteroid Ceres. I mean, in general, what we would probably want is something which is more like species, comma. I mean, this thing ultimately will be something that is some kind of species, chromosome. Okay, another biology question. Is it the case? Okay, let me just ask this question. Okay, is that a correct specifier for a particular base pair in any known living thing, or is that not a if the species specifier, like presumably there's a like in NCBI, there are discrete species even when they're viruses and bacteria. Is that a true statement, or is that not a? Is that some designator?
Sure.
Go ahead.
There is an NCBI code, a taxonomic ID for everything.
Okay.
People argue about the binning and so forth, but there at least is an official NCBI designation.
Okay, so there's a tax ID there. No, that doesn't. That's not. That's not. Okay, forget that. But anyway, there's something like that. And then, are there numbered chromosomes for everything in the known biological world, or does that not how it works?
For things that have been kind of yet described officially, yes, all the chromosomes will be numbered. Although, like some things, they'll be lettered instead rather than numbered, and you'll have chromosome HA and chromosome NP and so forth, just to complicate things. But each thing will have official chromosome. There'll be other taxa that exists that doesn't have official chromosomes to it at that point. But in that case, you couldn't do anything with it anyway.
Okay, but so like MT for the mitochondria and for the humans is an example of a named chromosome rather than a.
Yeah, that's good.
And then the strand that always exists, yeah, because it's two-stranded DNA type thing. Okay. Okay, so I reckon that we need a some kind of maybe we even genomic position or something that takes things like this, and where we can say things like one genomic position, maybe even minus another genomic position. I don't know whether that makes sense, but where we can do things like say, give us the biosequence that is sort of bounded by these two genomic positions. Is this at all sensible?
Yeah, take a brief look at. I pasted into at some point. So there's a concept of beacons now that human geneticists have been working on, where they're trying to, because this isn't a novel problem. Everyone has this thing where like we can't figure out if we're talking about the same thing, and so there's been work on the human genetic side to try to have this concept where you can actually have an agreement that yes, this location is the same location.
Okay, so in other words, this is like, so in the case of geoposition, we have this notion of datums for the Earth, because there are different, like the North American datum is different from the GPS datum, blah, blah, blah. Is there going to end up being something similar to that? I mean, there must be that for the reference sequences and things.
That's right. At the very least, we're going to have to also include a builder assembly number, so we know.
Yeah. Yes.
Okay, fine.
Yes.
So I'm taking seriously this genomic position or whatever it is.
I'm excited about it too, actually.
Right. I don't know if that's quite the right name, but it's something like that. And then that becomes, in a sense, then it's it's almost like geographics, where you can say, show me the stuff, show me stuff right around that genomic position would be probably a useful thing. I'm not sure exactly how that works.
Should it be slightly more general so that it also applies, say, to proteins?
Yes. So you're saying biosequence position or bioposition or something. So maybe biosequence position, and then it's got to have a specifier of. But in a sense, this is, this is saying, you can have a biosequence that is completely synthetic and just, random molecule, but this is grounding it to a thing that evolution has actually produced, or that people have actually identified with taxon IDs and things like this.
So the first argument could be an explicit sequence, then you can stick in whatever you want.
That's fair. Yeah, that's fair. So, I mean, this needs some work about what this should look like. Okay, fine. So let's keep going here. So that would make this look a lot nicer. Now, I guess this business with positions, again, I think this should work the same way as text position works. Okay, we've got a lot to look at here. So let's just do a quick scan through all of this. I unfortunately have a time constraint in about 20 minutes or so. the
This is given what's found, then extract the subsequences associated with it.
Okay, I'm going to make a meta point about all of this. This is clearly a large subsystem. I think Michael has already made this point. I mean, this is going to be a lot of work. It's going to be great, but it's going to be a lot of work. Okay. Trying to establish. What is this? Should evaluating protein entities result in a list of biosequences? So these are like proteins, like, I don't know, that assemble themselves in little, pentamers and things. Is that right? Or is that the...
Where there are multiple sequences, really, that make up the protein. So
right, and where it has some kind of self-assembly thingy or something.
Right.
Is that true? Or, I'm thinking like the capsids of... I don't know if I know what I'm talking about, but I'm just curious whether this is what you mean. Like the capsids of viruses. Is that an example of something like this? I'm hoping somebody like Trevor will help me here. And how do those work? Yeah. Those are many proteins, right? That self-assemble into the actual shape.
Yes, like the polymerase complex.
Okay, and that's what we're talking about here. Is having, is that right? Or in other words, when you have a protein, and
yeah,
it can have multiple biosequences, but then there's a higher level of structural organization that goes beyond just the sequences, because you have to say how the thing arranges.
That's right.
You could have a structure in protein data bank that to make that structure, you need multiple sequences.
I have to say that I think that we're going to need eventually something. Okay, so there's a question also for the chemistry folk, right? So you've got things which are molecules. Okay, more naive questions. You got a molecule, it's a covalently bonded thing, and it hangs out with, through some, I don't know, hydrogen bonding, or I don't know what, some weaker bonding thing. It's a cluster of stuff. How do you represent that? How are we imagining representing clusters? That's a question for Jason.
Right. So right now we don't have any non-covalent bonds. We've talked about having a hydrogen bond and other types of bonds. But as far as a cluster of molecules weakly bound together, we don't have any design for that right now.
Okay, so science question. In these things like these, these multimers in the bio case, what is the binding between the different proteins?
I should know that, but I can't tell you.
Okay. Are we talking about magnitude of strength, or do you want the type of bond that's holding them together? Because they're not covalent.
Okay. So the protein itself is held together covalently, but when they form these multimers, they don't. Those are just some different kind of bonding. Is that right?
I think you guys are looking for quaternary structure there.
That's right.
Yeah, we have that problem too. And even when you have two nucleic acids just hybridized, Watson-Crick paired to each other versus one, you have to represent them as different things for us. So we created a thing called like a structure, which is a higher level join of, I guess, a biosequence and a biosequence bound to each other.
Okay, that's what I was guessing we're going to have to do too. So so it becomes something like a biostructure, which contains biosequences.
If biosequence contains the structural nesting on the inside and can represent multiple sequences, then all of the existing functions for that operate on biosequences don't necessarily need to be redefined. We had, I guess, an earlier design that we didn't quite figure out all the details of, which would say that you have biosequence contains multiple biosequences, and then there's some meta information with how they're
okay. But let me ask a simple question. Okay, so we've got some RNA, and we actually can figure out the tertiary, the spatial layout of the RNA. Probably, I don't know. I gather that's decently doable.
Yes, definitely.
So now in our biosequence, a little bit like in the molecule case, right? A molecule could just be the kind of chemical structure, but it could also have the spatial information. Does biosequence also, in your design, allow some specification of spatial information?
Yes, there's a lot of room for meta information and additional options to specify structure, origin, and so forth.
Okay, so what you're saying is, okay, whatever we do here should be similar between molecule and biosequence for dealing with these clustery type things. So in other words, if molecule is going to have multiple sort of chemical units inside it, and because presumably with a molecule, if we have some cluster, it's specified by the coordinates of things in the different. What's the difference? I mean, yeah. Well, do we have okay? In the case of molecule, do we have a current thought for how we're going to handle even the ionic bonded things? What are we doing for ionically bonded things?
So let's see. So the 3D structures we come up with with ionically bonded they're not let's see, they're not good, right? So we just lay out each piece.
I know
separately.
that, and it's a big mess because you could have a crystal, and I don't know how we represent the crystal.
Right. I mean, so with crystals or whatnot, I envision some different head, like some type of a unit cell type of function.
All right, let's ignore the infinitely repeating ones, because that's and presumably, okay, naive question, like in the lipid bilayer thing of a cell, do you have, okay, in biology, do you have infinitely repeating things like that? Are they relevant, or is it only these information carrying sequences that anybody cares about?
For this project, we've really only attended to the information carrying ones, but there are certainly infinitely repeating, structures that are of concern for, like we say, for lipid bilayers, and then
and what about
other lipids.
What about in the case of ordinary molecules when we have polymers, like PVC or something, or some other? I don't know. Are there polymers that are considered infinite for purposes of chemistry, or is it always it's some specific length hydrocarbon or something?
No, I mean you can definitely, for all intents and purposes, consider them infinite. And I've always imagined that we would work our way around to having a macromolecule head that would allow us some type of in-between representation, halfway between the all-atom case and the biosequence case.
Okay.
Yeah.
Okay. Anyway, so one thing we clearly got to think about is how to deal with this, additional cluster type stuff for both the biosequence case and the molecule case.
Not to throw in complexity, but you've got to deal with some nonlinear stuff too. Like if you get to carbohydrates, you have to deal with branching and that sort of thing in the biosequence space.
Yep.
I don't know if that's macromolecule, but similar problem.
Yep.
Yes. And I mean, clearly with our ordinary molecule, we deal with that with having a graph, an underlying graph to represent where the component, how the components are connected to each other. But is that not scalable to the macromolecule case, or is that still, is it what you would do is to represent, and I think this was similar to what you were saying about multiple biosequences, where you have each linear chain stretch is one thing, and then you're basically just saying, this is how those linear chains are connected together. Here's the graph of how they're connected. Is that a reasonable way to think about it?
That's definitely how I think about the macromolecule. So you define each individual unit and whatever hairs it has coming off of it, and then you construct the graph from those. And if you have repeating units, then you are saving space by doing it this way.
Right. Brian, does that seem reasonable to you based on what you've seen with these sort of more exotic carbohydrates and so on?
Yeah, definitely. In fact, if you want to look up good precedents there, you should look up the representations people use, especially in the carbohydrate databases and that sort of thing, because they have to handle the branch structure as a regular thing, and you don't want to draw the full atomic structure out for something like that when you're just saying it's this sugar branched to this sugar at this position.
Okay, science question. How many levels of branching are relevant in your average, what we eat carbohydrate type thing? Is it like 1 or 2, or is it like many?
Excellent question. I don't think it's that crazy. I think it's less than double digits.
Yeah, I think it's 5, 6 maybe. Like in the comment.
Yeah, 5 or 6 is not that unusual.
And what's the thing what's the example?
Just I guess if you had a glycoprotein, you might have a chain sticking off the end of a protein, by the way, that then goes into the carbohydrate. That itself might have something like 5 levels of branching, not much more than that.
Fair enough. Okay. Existing functionality. Oh my gosh. There's a lot of stuff here. Yeah, I mean, I don't know whether this representation, I've, in my little adventures with my own genome and so on, this representation is not horrible. I don't know whether, given that we're taking this stuff more seriously now, whether we should think about, okay, the real question is, when you're going to use the thing, is this representation useful? Right? So it's like you could imagine, one of our very early successful sort of kind of more implicit representations was interpolating function. Question is, when you use one of these sequence alignments, how do you use it? What do you do? What operations do you do on the sequence alignment?
So usually when I work with sequence alignments, it comes out of something like muscle or MAF or whatever. And there I get a FASTA file that is just going to be string of the reference, string of entity 1, string of entity 2, string of entity 3, and each string will have dashes inserted into it. And so now they total the same length, and I can now just do simple for loops and so forth and work through them, where if I want to work with column 1, I can just iterate through column 1, column 2, and then look at diversity and be able to work with the
I see. So one thing you're saying is that we need an operation that will take, after sequence alignment has been done, that will take something like this and return the individual strings with dashes to represent where, like in this case here, it's not totally obvious to me that there isn't necessarily a unique place to put the dashes. Is that true? Or is there always, if it's a deletion, yeah, I guess there is. Is that true if there's more than 2?
There might be, there will be, for this particular alignment, there will be a particular dashing set up for it, or there could have been multiple ways to do an alignment that are optimal.
I see what you're saying, right? It's the minimal diff. The alignment represents the diffs effectively, and the diffs might not be unique.
Exactly.
That is right. Okay. But so one thing that's clearly useful, I can see why that would be useful, is to just take something where you turn this into a thing. Do we have that operation, John, of turning this into something with equal length strings with dashes in them?
We don't have that, but that's great. That'll handle multiple sequence alignment a lot better, and that yeah, I'll look into that.
Okay. All right. Let's just oh my gosh. Okay, I'm going to open a bunch of these just because we're running out of time and I want to see what's here. Okay. Okay, that's pretty straightforward. Yeah, the challenge here is, are we doing this with sub-value properties, or are we doing it with functions? And I kind of agree that we should have some functions. It just scares me when I see that number of functions.
It feels more natural because you do something on the thing. It's not you have the thing and take something out of it.
No, I agree. I agree. Okay. Obviously, the transcription target. I would say this transcribe is specific. What about from RNA to protein? Is that a?
Oh, that's translation. Yeah.
Oh, fair enough. Oh gosh, that's a can of worms, isn't it? Oh my gosh. So, okay, fair enough. Offsets and frame adjustments. Oh my gosh, what a can of worms. Okay, so that's essentially, this is analogous to when we do binary read list. I mean, it's kind of outrageous, but it has to be that way. This thing about read to a terminator and so on.
Right. Here we're reading to a start codon or
okay, so we already have a syntactic mechanism. I don't know how good it is for like reading to a new line and so on. We probably should look at that to see how that corresponds to this. I doubt that we're going to want this whole, given that there's a very specific function, biosequence translate, I don't see why we need an option here to say to start codon. That's more like an argument here.
Oh yeah, that's fair. The one thing that's tricky about that is that these options also apply to the inverse translation. And the inverse translation, you can either take a sample with a single argument, or you can have all translations by adding all as a second argument. So which argument, there will be some design to fit.
Yeah, okay, fair enough. It does have to be. Okay, fine. Okay. What is this? Genetic translation table. Oh, I see. Oh boy, that's this business.
Right. It's to handle all the archaea and.
How many different cases? How many different standards? I wouldn't say ISO standards, but how many standards has evolution come up with for how this translation works?
Yeah, there are about 30 standardized ones.
Wow. Of exotic amino acids that exist only in,
no, just of particular translations. So the codons are standard. The amino acids are standard here, and then it's just different.
And it's all kind of degeneracy offshoots of the one master genetic code. It's not like multiple kind of parallel.
That's true. Yeah.
But so when you have a degenerate sequence, genome sequence, what happens when you say translation? Does it? How can it know what to do?
So it will. That's why I guess the things have the option of either one or all, and why this is I guess an option here. So if you just say translate, it picks you an arbitrary one. If you say all, it will give you back a list of all of them.
A little bit messy. That's the difference between replace and replace list, so to speak. In other words, is it just a single? Yeah. Okay. Oh boy. Okay. I don't even know what that is at all.
So these are two sequences, and this is finding the degenerate sequence that covers both of them.
All right, let's have a quick look ahead here.
So these are things this entire section are things we don't include, but are just considerations for future functionality.
That's an interesting issue, because obviously you can't do comparisons if you've put UUIDs in the middle of things. On the other hand, there may be cases, and Emerald folk probably have opinions about this, there may be cases where it is really good to have, or the question is the UUID might be in an outer layer that, I'm sure Emerald has UUIDs for everything, but probably not down at this level. In other words, you have a wrapper, you have a container, that has a UUID, but it's not clear the thing that is contained has to have its own burnt-in UUID. Do you have an opinion about that?
We have a Constellation, which is our database, looks a lot like the entity system, but with fixed ontologies for everything. So you just type it as, this is what's an example?
Just like we have we have IDs for all of our objects. So like a piece of analysis you did has an ID.
This is a strand model.
Fair enough, right. So everything that goes in your database has an ID.
Yes.
Right, and for us it travels, right? So you could put it into the DNA synthesizer and say, go make this thing. And it knows it without having to transmit everything along with it.
Yeah, I understand. But that's exactly kind of what I was saying, that the wrapper contains the ID, rather than the wrappee, so to speak, containing the ID.
Yeah.
Yes.
And I kind of think that our wrappee should not contain the ID any more than the number 13 contains an ID, so to speak. In other words, you just do whatever you do with 13, but if it needed to be wrapped, then that will be my initial take.
I agree.
The case that is for are things like the short read mapping, where you have a large number of very small reads coming up and you're mapping them to specific genetic features. And at the end of the analysis, one analysis comes back that's the entire alignment, and you want to see, okay, where did each of these reads end up? And so it might not be more of a, this might not be a general thing.
That's a different issue. That's a different issue. I mean, that's kind of the issue that we're seeing in I don't know, something like face recognition or something, where you've got an image and it's like, these are the faces in the image. Okay, where are those faces in the image, and things like that.
Right.
And that we've got different patterns for dealing with. By the way, I have a question with respect to the actual processing of data. I mean, to what extent are we going to be in a position, if people have fancy special purpose, I don't even know these days what is the, surely there's GPU code for dealing with sequence and alignment. What will be our story in terms of being able to make use of that? Or is it not the case? I mean, I would think that was isn't. I mean, I don't really know what is the state of the art these days for. I mean,
I use math for most of my sequence alignment. It's not GPU enabled, but it does handle things pretty well. Yeah, we talked about this. I talked about this, John, previously. Basically, where the way that most of the stuff in the lab happens now is use things like SnakeMake and Nextflow to string together a lot of different scripts, and like some of those might be our own scripts, and some of those might be running math and running other things. And then I picture a lot of this being useful is being able to quickly, you want to do I/O to kind of pull things into from FASTA or from whatever format it is, process something within Wolfram, and then be able to export an artifact that can then be kind of inserted into our data flows.
And then the question is, what formats do those existing data flows eat?
For what we do, FASTA and VCF and
yeah,
BAM
those are long-running formats. I mean, those have been around a long time.
Exactly.
Okay. And are there new ones that are sort of?
The main new ones that are new within the last maybe 10 years are things like BAM, which are just designed to be shorter ways to represent lots of genetic data. So you don't represent all the sequences. Instead, you have a reference and you just say what's changed versus the reference.
Fair enough. Okay, so these are things for machine learning basis for, let's see.
Generally speaking, yes.
Okay. And this whole business about visualization, which seems important, to have some generic, to have some standard collection of biosequence visualizing tools. I mean,
that's right.
But I mean, is it the case? I mean, look, in my own little efforts poking around in my own genome, I have never used these interactive sort of exploration things. They're very cool, but they're not very useful to me, at least looking for particular things. What is the typical experience? I mean, these things which are sort of genome, genome explorers that are very interactive. Are they viewed as being highly desirable or not?
I know that Isabel's made a fair, in her lab work, has made a fair use of particularly circular views where she's trying to determine, where to insert things in plasmids and things like this. But it's particularly good for things that are naturally.
Plasmids are small, aren't they? Usually.
Yes.
But I'm asking, like the human genome viewers, where you're doing the giant zoom from infinity, and
right.
Is that useful, or is that just a toy?
For me, that's largely a toy, but the kind of the corollary where you actually have experimental data from RNA-seq or some other sequencing experiments that happen very commonly, just getting that read stack where you can kind of see the reference and you see how your million reads stack onto it, and you can kind of have the very zoomed out view where you just kind of get little, like almost sparkline little kind of, you see where in the genome's hit, and you can zoom in and see like, oh, we're getting SNPs in this position and so forth. That is the most common kind of interactive.
Right, and I mean, we haven't even discussed that, the question of the actual stack of reads. And, we're talking here about consensus genomes, aren't we? And, there's a question, because that seems like a useful thing that people actually care about, is because one of those things was an undetermined base. You actually care what the stack of reads at that position was. Is that true?
That's right.
I really have to run, unfortunately. I wish we could have gone on with this, and another time we should schedule it for longer. This is a lot of stuff to go through. This is very helpful as an introduction. This looks quite wonderful. I mean, this is going to be a great system. It just it needs work.
True.
We knew that.
We only came to this point by doing already a fair amount of work.
Yes, I can see that. Right. But this is yeah.
You want another 2-hour meeting?
Let's do a 2-hour meeting. Yeah, with as many people from here as can attend would be wonderful.
Yeah, happy to reconvene.
Okay. Well, thank you all very much, and thanks.
Thanks.
This is looking wonderful. All right, I got to run, unfortunately. See you all soon.
Thanks. Have a great day.
Thanks. Bye.